Hi everyone. In this video, we're going to talk about protein structure. How does a protein get its shape, for instance? So there are four different types of structures for a protein. There's the primary structure, the secondary structure, the tertiary structure, and finally the quaternary structure. So let's just step through each type of structure. So the primary protein structure is just the linear sequence of amino acids in a protein chain. So if you took a protein and unfolded it and stretched it out, that would be the primary structure. We're just looking at the sequence of amino acids. And the sequence of amino acids will determine further structures, which we'll talk about in a second. So off to the side, we have an example of a protein that's kind of been unfolded. It has two chains. One is called an alpha chain, one is called a beta chain. And here we just have the sequence of amino acids written out for each chain. So we've got, for instance, phenylalanine next to valine, next to asparagine, and we can keep going. And the sequence of amino acids is really important for the function of proteins. So if you have small variations in the primary structure, so in the sequence of amino acids, that can cause a lot of differences in how the protein folds and then how it functions. So having that sequence in the right order is very important. You can also see, for instance, there are some disulfide bridges here between cysteine amino acid residues on each chain, and that's going to help with the shape of the protein as well. So if, for instance, we didn't have a cysteine here or here, that could greatly affect the shape of the protein and how it functions. Okay, so also when we stretch out that chain and just look at the primary structure, remember that we have these different amino acids that are all linked by a peptide linkage. So I'll just kind of draw a few of these. So we've got amino acid 1, 2, 3, and they're forming these amide bonds, these peptide linkages as we go down the chain. So keep that in mind because the next thing we're going to talk about is the secondary protein structure. So the secondary protein structure is determined by hydrogen bonding between amide groups of amino acid residues in the chain. So let's go back. What's going to happen here, actually I'll use green. So the carbonyl group of one amino acid is going to form a hydrogen bond with the hydrogen on a nitrogen that's 4 amino acids away. I think I did that right. So we're going to form a hydrogen bond between the carbonyl group of one amino acid and then the nitrogen on another amino acid. So the amide groups are forming a hydrogen bond. And you can see that here as well. We've got the oh, I did not mean to do that. I meant to highlight it. So the carbonyl group of one amide in an amino acid is going to hydrogen bond with the hydrogen on the nitrogen of another amide group on another amino acid. So what ends up happening is you have either an alpha helix that occurs or a beta pleated sheet. So an alpha helix occurs when a single protein chain twists into kind of like a curly Q, or it can also resemble a coiled helical spring. And this is where hydrogen bonding occurs between carbonyl and amide every 4 residues. So let's take a closer look at that. All right, so this is an alpha helix shown on the right. And alpha helices are found in a lot of proteins. Some of those proteins are listed here, like keratin, myosin, epidermin, and fibrin. Most of these are structural proteins, but again, notice that we've got our primary structure, which is just the order of amino acids going down the chain. And then we have our secondary structure, and that's formed by the hydrogen bonds between amide groups, and this occurs every 4 amino acids. So that's kind of cool that just these hydrogen bonds can form a little curlicue in our protein. Now the other structure that you can get here is called a beta pleated sheet. Oops. So that occurs when several protein chains lie side by side. So they're going to be held together also by hydrogen bonds between the amide carbonyl oxygen atoms of one chain and the amide hydrogen atoms of an adjacent chain. So again, with the alpha helix, we just have one chain that's curling in on itself. But with a beta pleated sheet, we have multiple chains that are lying side by side. So let's look at how a beta pleated sheet forms. Okay, so here we have 3 different chains. We've got one on the left, one in the middle, and one on the right. And they're lying side by side, and they're lined up so that the carbonyl group, CO, on one chain forms a hydrogen bond with the hydrogen on the amide group of another chain. And you can see that this occurs multiple times. And this is what's kind of gluing the chains together. Now notice that we have this pleated sheet that forms, and that's just due to the geometry of the atoms. Most of them are either in a trigonal planar geometry or a tetrahedral geometry. And that's going to create that pleated sheet structure. All right. So now that's going to bring us, actually first, before we go to the next type of structure, some proteins can actually contain both an alpha helix and a beta pleated sheet. It just depends on how it folds. So in this example, the protein chain is wrapping around, and at one point, the amino acids will kind of line up in such a way that we get this coil. But then further down the line, it might line up with another or different section of the protein and form a beta pleated sheet. So it really just depends on the order of amino acids, and then if they kind of line up in the right way, or if you get those hydrogen bonds lining up just right. So again, if you have a beta pleated sheet or an alpha helix, this is part of the secondary protein structure. Now that brings us to the tertiary protein structure. So the tertiary structure is the specific 3D shape of a protein. And this will result from interactions between R groups, or the side chains of amino acid residues. So side chains, remember, can be divided up into 4 different types. We have nonpolar, polar, and then we had basic and acidic side chains. So the types of interactions we could have are disulfide bridges between cysteine amino acid residues. And cysteine is the only amino acid that could form a disulfide bridge. And then we could have salt bridges, which are ionic bonds that form between acidic and basic residues. So we'll look at an example in a bit. You could also have hydrogen bonds between the side chains, and that would form between polar side chains on amino acid residues. And then hydrophobic interactions, and that would form between the non-polar side chains of amino acid residues. Okay, so let's look at a picture of that. So if you have a protein that's kind of randomly, folded up, it doesn't necessarily form an alpha helix or a beta sheet. That will be due to those four different things. You might have just disulfide bridges, you might have just hydrophobic interactions, or you could have a combination of all of these different types of interactions. So here we have an example of each type. So we've got our disulfide bridge, and again, that would only form between cysteine amino acid residues. We could have an acidic amino acid that then lines up with the side chain of a basic amino acid, and then the negative charge on the acidic side chain would be attracted to the positive charge on the basic side chain, and they form a salt bridge. So remember, a salt is something like sodium chloride, where you have a positive sodium and a negative chloride. And this is the same thing here. You could also have hydrophobic interactions. So if the side chain of the amino acid only has carbons and hydrogens, or it's relatively non-polar, then those could be attracted to each other. So for example, these are benzene rings, and those only have carbons and hydrogens, and those will be attracted to each other mostly because we're typically in an aqueous environment, and anything that's non-polar will not dissolve in water, right? Because water is polar. So those non-polar side chains want to get closer together to get further away from water. And then finally, we could have, for instance, a polar side chain, like an alcohol group, and that would form a hydrogen bond, or those two side chains would form a hydrogen bond. So these are all the different ways that we get a tertiary structure. And again, that has to do with interactions between the side chains, the R groups. So in an aqueous environment, again, we have hydrophobic side chains, so non-polar side chains. And then we have hydrophilic side chains, so they could be polar side chains or side chains that have a positive or negative charge, so acidic or basic side chains. So that can also determine the tertiary structure. For instance, we could end up with a globular protein due to these different interactions with water. So in the middle, you might have side chains coming off of amino acids that are all non-polar, and they want to face towards each other so that they can get as far away from water as possible, because non-polar molecules don't dissolve in water. But if you have anything that's polar or has a positive or negative charge, those can face outward towards water because they're attracted to water. So now we come to the last type of protein structure, which is called the quaternary protein structure. So far we've covered primary, secondary, and tertiary structures. All proteins have these three structures. They just kind of build on each other, right? But not all proteins have this quaternary structure. So quaternary structure is the arrangement of subunits that form a larger protein. So one really good example is hemoglobin, which we saw before. And remember, hemoglobin has 4 different parts to it. So those are its subunits, and they're all kind of bound together to create a larger protein. So that would be the quaternary structure. So the subunits that make up the quaternary structure of a protein are polypeptides that have primary, secondary, and tertiary structure. They might be identical or they might be different. It just depends on the protein. Now, some proteins are conjugated, so remember that means that they have prosthetic groups. So conjugated proteins with quaternary structure, again, contain these prosthetic groups. So again, with hemoglobin, each piece of hemoglobin or each subunit had a heme group, which is shown here, and that is a prosthetic group. And then here's a slide just kind of showing what we already talked about with hemoglobin. So again, there's 4 subunits. 2 of them are identical alpha chains, and then 2 are identical beta chains. So each subunit has its own heme group. So there's a total of 4 heme groups. And what's actually holding those 4 structures together are hydrophobic forces. Okay, so let's review everything we've gone over so far. So what I would like you to do is complete each of these sentences. So the primary structure of a protein is ... The secondary structure of a protein is ... etc. So feel free to go back in your notes and complete these sentences. You can pause the video while you do that. And when you're done, you can unpause and we'll go over them together. Okay, so let's start with the primary structure. So the primary structure of a protein is the linear sequence of amino acid residues. Okay, simple enough. So then the secondary structure depends on the primary structure, but the secondary structure of a protein is determined by H bonding, hydrogen bonding, between amide groups of amino acid residues. Residues, there we go. So remember, there's 2 types of structures that could form here. You could get an alpha helix or a beta sheet, a beta pleated sheet if you want to use the full name. All right, the next one, the tertiary structure of a protein is the 3D structure resulting from interactions between side chains or R groups of amino acids. And remember, there's 4 different types of interactions that could occur between the side chains. So you could have disulfide bridges, salt bridges, hydrogen bonding, or hydrophobic interactions. So you could also write that down if you would like in your notes. I just ran out of room on this slide. Okay, so the quaternary structure of a protein is the arrangement of subunits that form a larger protein. Okay, so I would like you to know these 4 definitions, or you could even rewrite them in your own words. You don't have to memorize them word for word, but as long as you have the general idea of each type of structure, that's the important part. Now, one more thing before we go, protein hydrolysis. So we've talked about how proteins build up their structure, but proteins can also be broken apart. And oftentimes, once a protein kind of does what it needs to do, it will be broken apart into its individual amino acids, and we can then use those to build other proteins or other structures. So there are different ways to break apart a protein. You can use heat, acids or bases. So for instance, if you had a protein and water, you can hydrolyze it, but you'll probably need an acid or a base to catalyze the reaction. And that will break the protein up into smaller peptides. And then you could further divide those peptides up into their specific amino acids using, again, acid or base or heat. So this is a really important process in protein digestion, and this uses enzymes to help catalyze the reaction typically. There's also protein denaturation. So this is where a protein loses its native state. So the native state is what we use to refer to all of those structural elements we just talked about. So the quaternary, tertiary, and secondary structure, but not the primary structure. So for example, this is similar to eggs. When you crack an egg into a pan and you cook it, you're using heat to denature it. And then you could further scramble it up, etc. So denatured proteins, you've got your kind of structure that's broken down and it no longer functions the way that it did before. So again, there are many different ways that you could denature a protein. You could use heat or ultraviolet light, and that will disrupt hydrogen bonds, so it will break apart any hydrogen bonds or ionic attractions by making the molecules vibrate too violently. So this would produce something called coagulation, as in cooking an egg, which I just mentioned. You could also use organic solvents like alcohols or other substances that mix with water. And this will also disrupt hydrogen bonds. You could use strong acids or bases, which will also disrupt those hydrogen bonds and ionic attractions. Now, if you expose a protein for a really long time to acids and bases, eventually you'll get hydrolysis of the protein where you get those individual amino acids. You could also use detergents, which will do a similar thing, or heavy metal ions like mercury, silver, or lead. So mercury, silver, and lead will form bonds to any thiol groups, which are those SH groups. So for instance, cysteine has that SH group that we saw. So that will break apart any disulfide bridges. And then it will also precipitate proteins as insoluble heavy metal salts. So they'll no longer dissolve in aqueous solutions. So there's a lot of different ways that you could disrupt a protein's structure. Okay, so that's it for proteins. Well, except for the next chapter, we're going to talk about enzymes, which are a type of protein. So we'll try to kind of wrap up the enzyme chapter a little more quickly than the protein chapter. But I will see you in the next video.